Good morning, ladies and gentlemen. My name is Ansingnaa Desmond, and I'm presenting on microbial bacterial contamination of spectacles. Bacteria, as we all know, are microorganisms that are seen only under the microscope. These organisms are found everywhere in the environment. That is, as human beings, every day we come into contact with these bacteria, and some of these bacteria are harmful while others are useful to the human system. Bacteria are found to have inhabited a variety of environments. That is, they are found in so many areas within the human environment. And a study that has been carried out shows that the Ghanaian city has been found to contain some bacteria that could be pathogenic. And also, it's been found out that the lab coats that people wear, that is by doctors or by lab technicians or even by us as students when we come for practicals, these lab coats are meant to protect us, or they are meant to serve as a protection against some of these things, but it has been found out that these lab coats also harbor some bacteria in them. And recently in the UK, there is a research that was carried out among surgeons, and the spectacles that surgeons wear have been found to also harbor some bacteria which are pathogenic. And since the research was carried out in a hospital base, it was inconclusive as to whether the research, the contamination was due to the hospital setting or it was as a result of what? Mishandling of the spectacles by the doctors. It is based on this that we therefore there's the need for us to find out whether there is a possible contamination of the public spectacles, that is spectacles that are used by the public. Since the project that was done was carried out in a hospital setting, we couldn't just conclude that the same bacterial contamination will also be on the spectacles that are used by the public. Hence, the need to carry out this study. It is necessary to carry out because once the study is being carried out, information will be made available to help improve on the use of spectacles. That is, it will go a long way to prevent infection that may result from this bacteria. Since bacteria are opportunistic, they can easily infect the eye. And once the spectacles are worn very close to the eyes, if the bacteria are harbored in the spectacles, they can easily get infected, or they can easily give infections to the ear or the eyes. And then also it will give information to the prescribers, that is, as to how they inform their clients how to be able to use the spectacles or handle them very well. This study is aimed at possibly determining the bacterial contamination of the spectacles. But specifically, we'll be looking at the types of bacteria that are harbored, that is by the spectacles that are used by the general public, and also the clinical importance of such bacteria, as whether they are pathogenic or they are just normal microbial flora. It will also ascertain the possible care practices that are carried out on the spectacles that people use, since some people may not use them or handle them very well. We therefore hypothesize that spectacles harbor pathogenic bacteria. In this study, 100 participants are going to be recruited who might have been using spectacles for not less than 6 months, and also these people might or should have worn or should be wearing the spectacles every day for not less than 5 hours a day. So during the study, swabs will be taken from the spectacles. That is, from 3 places on the spectacles. It will be taken from the lens itself, the nose knob, and then the lens, the temples, that is the handle. And as a control, new spectacles, the spectacles that are not being used will also, the swab will be taken from the same places and also project to find out whether the bacteria that will be found on the spectacles used by people, whether the contamination is as a result of their improper handling or is as a result of the manufacturing company. The swabs will be cultured on a chocolate agar, and this agar is a non-differential agar, and to be able to show or any bacteria that will possibly be found on the swabs will be able to grow on such an agar, and then they will be sub-cultured on a McConkey agar, that is a differential agar, that will be able to differentiate the different strains of bacteria that will be found. questions or questionnaires will also be administered to the participants. That is to ascertain the possible care practices that they make on the spartacles. That is how they treat them, how they clean it, how many times, and what do they use in even cleaning these spartacles. The results will be analyzed using SPSS, that is the one-way ANOVA. It is expected that there might be bacterial growth in the cultures that will be done. And then the bacteria that are expected, we also expect to find pathogenic bacteria there, because this may be a contributing factor to to worsening people's health conditions, even as some people complain when they wear spectacles. Application of the results, that is, it will add knowledge to the already existing knowledge that we have, and also it will give an information to the manufacturing companies. That is, if it is found out that the new spectacles that the swab will be taken out from, they also contain some pathogenic bacteria, this will inform the manufacturing companies as to how to manufacture the spectacles or the antibiotics they should use in treating their materials. It will also inform prescribers. That is, as the doctor prescribes the lenses to the clients, it will give the information on how they will be able to tell the clients to handle their spectacles very well. And also, it will make available information to the general public as to how to handle their spectacles, how to clean them, and the other things. This is my schedule for the project and my budget. Thank you. The 100 participants will be recruited from the Department of Optometry, that is the clinic over there. They have the number of people, or the people that visit the clinic, those that come, they issue out glasses or spectacles to them. So from their records, we'll go through the records, and those that have used the spectacle for not less than 6 months, we'll get the records from that.
What kind of sampling technique is that? What kind of sampling technique is that?
It's a sampling by convenience.
Any other questions for Desmond? Why sampling by convenience?
Sampling by convenience because, as it is now, the first this that was done for only like 40 people. But this one we are looking at 100 people. If we take so many people into consideration, because it's just a project, it will be inconvenient. Like it will be to serve as a listen.
Have you already started taking samples?
No.
Then describe your convenient sample. Look at how you can randomly sample. Because where are you basing the 1,000 years? Is it on campus?
Yes, from the campus.
If it's campus based, then you can have a random sample.
You are doing the question for another target group. People have worn spectacles for 6 months and more, and have been wearing their spectacle or have been compliant at least 5 hours per day. You can't just go pick anybody else. You have to go for your target group before you can do that. Yes. So that's what you have to do.
The target group. The target group, that is sampling free. But then in the sampling free, you can still do random sampling.
You can. The issue is that it's more of an experimental kind of thing. Now we want to find out how they are cared, because for one spectacle, we are going to take swab from various parts, probably 3 parts. The temple, which are close or have some kind of attachment to the cheek, that is the skin. And then you have the nose part, this part. You see most of the people who have their own rose and kind of green nose. Another thing, you go and torture, do differentials. So if you want to say you want to do actual, let me say, random sampling, then it means that probably you have to get to the public. Get people who are wearing spectacles and the like. But now what we want to find out is that some people may not even be wearing their spectacles to even qualify for that particular thing. So we want people who are really using it. But that limiting factor is that we are thinking that probably the spectacle, the handling and the care by the wearers is what is contributing to the fact. So we have to go for
Yes, can you go to the application of the results? You were saying something that the new spectacle that we manufactured, right? From your supervisor, I heard that initially I was thinking that you also take the new spectacle and also take swaps from the new spectacles. I could do that.
Yes. That was as a control.
That was a control.
Yeah. That is, we look at like if the new spectacles that are, we will take the source from those ones too. So that if we find the same bacteria growth on the acid, the ones that people are using, then we can conclude that the bacterial contamination is not as a result of the people not handling the spectacles properly, but we will say that it is from the manufacturing end, where they are manufactured, that's where the bacteria are contaminated from.
Let's go to the virus.
Yeah.
I've never looked at it. Where's the current? Just bring the current out for your statement and then we see. I'm not seeing the current.
The problem here is that the bacteria that we found to have contaminated the spectacles of the surgeons.
Of surgeons?
Of surgeons, like orthopedic surgeons. That is
to do with surgeons, so I'm doing the normal population.
Yes, that's what I'm saying. Though it has been found out that there are bacterial contamination of this, the spectacles of the surgeons, there is no such information. They have not done any work on the general public. So that's why we deal with the general public to find out whether the same whether we will find the same possible contamination as was found on the spectacles of the surgeons.
Is there any other question?